Biomedical data can be complex and diverse, because they represent what we know about the state of a human organism, which is a complex system. It comprises cells with a nucleus containing chromosomes. The chromosomes contain genes made up of DNA, which code for proteins for the mediation of RNA. Proteins are responsible for many cellular functions, and so have a vital role to play in the health of cells, tissues, systems, and organisms. Broadly speaking, living organisms are made of cells, which is the smallest structural and functional unit of an organism. Some organisms have several cells, like human beings. Some have only one, like bacteria. Cells generally have a nucleus, in particular in humans. And in the case where the nucleus are also organelles is separated from the rest of the cell membranes, they are called eukaryotes. Otherwise they are called prokaryotes. For example, bacteria prokaryotes, because their components rest directly in the cytoplasm, without membrane separation and without separate nucleus. Cells are made up of different components, or organelles, such as a cytoplasm, which is gel-like liquid basing, the inside of a cell, nucleus, mitochondria, and also organelles such as ribosomes. Ultimately, cells and their components are made up of macromolecules, which are large aggregates of molecules. Talking about deep data, so you remember, it is made up of 37.2 trillion cells, as well as 40 trillion bacteria, most in the digestive tract, some say 10 to 1. It is called the microbiome. This is a complex system that exists many types of macromolecules and are more than 100,000 entries in databases of macromolecular structures. DNA, RNA, and proteins are examples of micromolecule composing cells. These micromolecules are created by living organisms. So what exactly is DNA? A little bit of biochemistry. Well, DNA stands for deoxyribonucleic acid. DNA stores the genetic material in chromosomes in each cell nucleus in humans. In viruses, for example, the DNA or RNA reside within the organism envelope, made of proteins without nucleus. There is also some DNA in mitochondria, called mitochondrial DNA. DNA is transcribed into RNA out of the nucleus, process called transcription. RNA stands for ribonucleic acid. RNA is translated into proteins in a cytoplasm organelle called a ribosome, process called translation. In summary, DNA generates RNA, which in turn generates proteins. DNA and RNA are composed of nucleotides, which are molecules of nucleic acid. A nucleotide is made of a nitrogen base, a molecule of sugar, and a phosphate group. There are two types of bases, the pyrimidines and the purines, and there are three pyrimidines: cytosine, present in DNA and RNA; thymine, present in DNA only; uracil, present in RNA only. And the other types of bases are purines. And there are two: adenine, A, present in DNA and RNA; guanine, G, present in DNA and RNA. Nucleotides are linked together through a 5-carbon sugar with phosphate, ribose for RNA and deoxyribose for DNA. So the main difference between DNA and RNA is that DNA can have cytosine, while RNA can have uracil. And they used different 5-carbon sugars. These are the three pyrimidine bases and their molecular structure made of carbon, hydrogen, and nitrogen. They have the shape of a hexagon. As you can see, different groups are attached to the core, such as here an oxygen methyl group or nitrogen group. And this is what different shapes between this pyrimidine bases, because you can see they have pretty much the same shape. And these are the two purine bases. More complex. They have a hexagonal core, and also pentagon, with different groups attached, such as oxygen here, nitrogen group, here some hydrogen, and These are called guanine and adenine. And these are the 5 carbon sugars that can be used in DNA. We see here deoxyribose and how it attaches to an adenine base to form a nucleic acid. There we see a deoxyadenilic acid made of adenine, 2-deoxyribose, and a phosphate group. The 5 carbon sugars serve as links between 2 nucleic acids. Here between an adenine and a cytosine. There is one extremity that is a 5' here, and one is that a 3', named after which carbon atom is involved. For example, here there is a 3'N and a 5'N and a 3'N, which is a 1. Having a phosphate group here. The group is not shown on this slide, but there would be in group. On the 3' end, succession of a nucleotide compose a single strand of DNA. What is beautiful is that from a simple sequence, extraordinary shapes are formed. 2 strands of DNA pairs themselves in the 3'D shape in a double helix, where bases are paired. We talk of BP for base pair. A pairs with T, and G pairs with C, and the pairings of the bases AT and GC provide chemical bonds responsible for the double helix shape. The 3D shape of the DNA takes the form of a chromosome, which tells the hereditary information about the person. The chromosome is a structure where the DNA is coiled around some proteins called histones. Chromosomes reside in the nucleus of cells. Humans have 46 chromosomes, 23 pairs. Chromosomes in 22 pairs look the same, but for the third, it's the gender chromosome. Males have an X and a Y chromosome, while females have 2 X chromosomes. See the karyotype to the right, which is a set of chromosomes in a human body. So chromosomes have this very particular shape of an X with a central mirror with 2 small arms and 2 large arms. They have a shape again, resembling an X. Now what is a gene? A gene is a part of the genome that can be transcribed to heal a protein. Actually, a gene may encode a protein through an RNA sequence, and genes are made of exons and introns. Exons are regions coding for proteins and are separated by non-coding regions called introns, while the coding regions are called exons. To simplify further details, genes are concentrated in certain regions of the genome rich in G and C. Regions reaching A and T do not contain genes. Between the 2 CPG islands, repetition of C and G, separate coding regions from non-coding ones, which are the introns. Non-coding regions can also be parts of genes. What I've said so far can vary greatly depending on the organism in terms of size and structure. There is a profound diversity of living organisms' genomes. For example, cells have DNA, while phages and viruses, which are acellular, can have DNA or RNA as their genetic material. So DNA can have a direction when it is double-stranded from 5' to 3' of the molecule. However, when it is single-stranded, it can be read in both directions. The genome can be organized, or not, in chromosomes. The human genome comprises 46 chromosomes, 3 billion bases, and 20 to 25,000 genes. It was thought to contain more, but the Human Genome Project completed much earlier than planned when fewer genes were discovered. Other species' genome vary in size and number of genes. Human genome has only twice as many genes than the primitive worm. However, onions, for example, have 12 times as much genome as we do. So the amount of genomic information can vary greatly between organisms. So GenBank database is like an encyclopedia of genetic information from over 100,000 distinct organisms.